Hello everyone, welcome back to my channel. Today we are going to learn about the different type of blood cells, one of the most fascinating topics in hematology. Blood is often called the river of life and its cellular components play a vital role in oxygen transport, defense and clotting. Blood is composed of two main parts. The liquid part of the blood is called the plasma and the formed elements which are the cellular component. The formed elements include three types of cells. The first one is red blood cells or RBCs, white blood cells or WBCs, platelets or thrombocytes. Let's start with the red blood cells, also called erythrocyte. They are the most numerous cells in the blood, give it the red color due to the presence of hemoglobin, and their main function is to carry oxygen from the lungs to the tissues and return carbon dioxide back to the lungs. So in this first picture, you can see mammalian RBC. How we will identify the mammalian RBC? The shape will be biconcave or circular disc, and the mature RBC having no nucleus. And the second picture is of an avian RBC. The shape is different from the mammalian. The shape is oval and it is nucleated. This purple color is the nucleus of this avian RBC. And another exception is of camel RBC. Camel RBC shape will be like avian, like either it is oval or elliptical, but they don't have a nucleus. Usually the camel RBCs are oval or elliptical. Mainly it is an adaptation for their desert condition. And mature RBCs in mammals lack a nucleus, so this camel's RBC also lack nucleus. And the lifespan of the RBCs is about 121 days, and the removal of the old RBCs happen in the spleen, often called the graveyard of the RBCs. Now let's discuss the white blood cells or leukocytes. They are the defense cells of the body, protecting against infections and foreign material. The WBCs are classified into two broad groups, granulocytes and agranulocytes, based on the presence or absence of the cytoplasmic granules. First, we will look into the granulocytes. Granulocytes include neutrophil, eosinophils and basophils. And the neutrophil are the most abundant WBC, and they are the first responders during infection and perform phagocytosis. And eosinophils, they are involved in allergic reaction and defense against parasites, and they have a very bright orange-red granules in stained smears. And the basophils are the least common and containing deep blue granules that is rich in histamine and heparin. They play a key role in inflammation and hypersensitivity reactions. So how we will differentiate these granulocytes? So first feature we have to look whether the nucleus, this purple color thing in the three pictures, those are the nucleus. So the nucleus of the granulocytes will be segmented. The segmentation can vary between cells. The neutrophils will have 3 to 5 segments, and the eosinophil have 2 to 3, and the basophil also having 2 to 3 segments. The second feature you have to look the presence of granules and the granule color. In the case of neutrophil, it won't take any type of stain, so the granules will be neutral. It won't take any stain, so the cytoplasm will be white in color, or its granules is white in color. And in the case of eosinophil, the granules will take the stain of eosin, so it will be bright red in color. And in the case of basophil, it take the blue stain, so the granules will be blue in color. And one more thing about the neutrophil, actually in the case of avians and rabbit, their granules take stain, so we can't call it as neutrophil. So in those species we call the neutrophil as heterophils. Next is a granulocyte. It includes lymphocytes and monocytes. Actually, lymphocytes are the central to the immune system. They include B cells, T cells and NK cells. And monocytes are the largest WBC. They migrate into tissue and then they are called as macrophages, which engulf pathogen and involved in tissue repair. Now we will learn how to differentiate between these two cells. So the lymphocyte, the nucleus is round in shape and it have a very less cytoplasm. But if you look into the monocyte, the size will be very big. See, these are the RBCs. Compared to the RBC, the size is very bigger cells, and the nucleus will either bean shaped or kidney shaped, or it will have some depressions, and it have very large cytoplasm when compared to the lymphocyte. Lymphocyte is a very small cell. See, compare the size with RBC, it is actually smaller than the neutrophil. And monocyte is a very big cells. Its nucleus is not, usually not round. It will have some depressions, or it will be bean shaped or kidney shaped. And cytoplasm, it have a very big cytoplasm, and cytoplasm can contain vacuoles because it is highly active phagocytic cells. Finally, let's look at platelets or thrombocytes. They are small disc-shaped cellular fragments derived from the megakaryocytes in the bone marrow, and their primary role is blood clotting. When the blood vessel is injured, platelet adhere to the damaged sites, form a temporary plug, and release substances that activate the clotting cascade. So here you can easily identify platelets. Usually in the case of mammals, the platelets are enucleated cytoplasmic fragments that is mainly formed from the megakaryocytes. And in the case of avian, this is a thrombocyte. In avian, we call it as a thrombocyte. It is a nucleated structure in the case of avians. So in this picture, you can see a granulocyte because the nucleus are segmented. And there are two types of granulocyte. Here, look at these cells. These all having a very clear cytoplasm, means their granules are not taking any type of stain. So it is a neutrophil. These and these all are neutrophil. But look into these central cells. Its cytoplasm is having some granules, and the granules are taking the stain red. So this is a eosinophils.